So it's my pleasure to introduce Oyageshio, who has been in my lab for about 6 years now. He's a graduate student in the population biology program, and he will be presenting to you 3 of the chapters that he's been working on. So I just want to highlight a couple of sort of amazing things about this project. We actually started this project on tuberculosis back in 2017, and then it got shut down during COVID, of course. So Oshi was really the first PhD student that we had pick up this project and look at the data, which of course means that he had to quality control huge amounts of freshly collected data. And then you'll hear also a little bit about the RNA sequencing part of the project as well. And I just want to emphasize again here, this part was really spearheaded by Oshi, and he wrote an NIH R21 grant, which was awarded for over $400,000. Extremely impressive. And then Oshi spent about 5 months in South Africa as well, working with Stellenbosch University, our collaborators, and performing field work. So he really knows the breadth of this project, you know, from the ground all the way through to the bioinformatics side. Yeah, please. So yes, without further ado.
Thank you so much for that. So today it's a pleasure to talk about what I've been working on here for the past 6 years, titled Why Some People Get Sick, Tuberculosis, Immunity, and African Genetic Diversity. So before we dive into the data, I want to start by talking about my personal experience. So I was born in Nigeria, and Nigeria has the highest worldwide incidence of malaria. So not only I was born with congenital malaria, I proceeded to have malaria over 15 times, at least 15 times in my life. I stopped counting at some point. But the thing that interested me and plagued me was that even within communities and households, some people would get sick and others would not. And so before I had the sort of language like host-pathogen interaction, genetic diversity, I had this working model that there are differences between people and that difference affects phenotype. In this case, malaria outcome. But this talk isn't about malaria, it's about a different pathogen, but the same question still remains. Why do some people get sick, in this case, active tuberculosis, while other people don't? And for this talk, all of my work for tuberculosis is going to be based in South Africa. So in tuberculosis, exposure does not guarantee an active TB disease state. So I'm going to walk you through a model of how that happens. And if there are any TB experts over Zoom, I know this is a simplified model, but this is not the TB talk. So here we can imagine this is an individual. The little ovals here is M tuberculosis. So when an individual is exposed, they could actually clear the infection immediately, but some people remain in this latent state. So they have a proportion of the bacteria residing in their lungs. So what I mean by latent TB is these individuals, even though they're infected, they're healthy, they're asymptomatic, they can't transmit it to other people, and you can be infected and die your whole life and you're fine. But about 5 to 10% of people that have this latent infection progress to active TB. And so by active TB, I mean you're showing really intense respiratory symptoms. So fever, coughing, night sweats, and it ends in death. It can end in death if left untreated. So it has about a 15 to 65% mortality rate. And currently our options in treating tuberculosis are very limited. So even though there's a vaccine, the effect wanes after adolescence, and the only cure for it is a 4 to 6 month course of antibiotics. So what is driving this progression from latent to active TB? So a little more facts about tuberculosis. It remains one of the world's deadliest infectious diseases, and it's caused by this bacterium M tuberculosis. 25% of the globe is latently infected. This is a huge number, but mostly in Southeast Asia and sub-Saharan Africa. and over 1 billion people die yearly from active tuberculosis. So zooming in on South Africa, it's ranked 10th in global TB burden, and 54% of people diagnosed with TB in South Africa are also living with HIV. So this is an important fact to know for the future. And people living with HIV are more likely to progress to active TB, and even during treatment, they're more likely to die than their HIV-negative counterparts. And other signatures of poverty, such as undernutrition and overcrowding, are also risk factors for tuberculosis. So to study this, my field site is in the Northern Cape province of South Africa. It's a high burden setting of TB, 926 per 100,000 people. And for context, in North America, it's like 2 per 100,000 people. So this is a really high incidence. And interestingly, it has the second lowest HIV burden in South Africa. Despite HIV being a high risk factor, this has really low HIV. And it's also a low density province. These are some pictures I took in Uppington during my time here, 2023. And majority of our study participants have Khoisan ancestry. So they are descendants of these hunter-gatherer groups in South Africa. So more context about that. These hunter-gatherer populations have a really deep population divergence from all other human populations. So about 150,000 years ago. And they also have really high genetic diversity. So when we think about a metric like heterozygosity, if you look across all the sites, people with Khoisan ancestry over in purple have the highest heterozygosity. And my PI, Brenna, and others have shown as the distance increases from Southern Africa, your heterozygosity decreases. And so this region, South Africa, has a really interesting and complex migrational history. So there was this pastoralist diffusion of pastoralists from Eastern Africa to Southern Africa 2,000 to 3,000 years ago. And 1,500 years ago, there was the Bantu expansion of people from Eastern Western Africa as well. And a couple hundred years ago, British and Dutch colonialists came over to Southern Africa as well. And with them, they brought indentured servants from Indonesia and India to work in South Africa as well. So we have this really complex history over several thousand years. And one signature we can see with that is admixture. And by admixture, I mean the populations have multiple ancestries that make up their genome. So over here, this bar plot, each bar is a clinic we've worked in, and it's color-coded by different ancestry groups. And we can see on average, 57% of the genome of the people we work with have this indigenous hunter-gatherer Khoisan ancestry, and the other ancestries reflect the migrational history. And so to summarize this part, we now know the Northern Cape has this unique mix of high tuberculosis, high genetic diversity, and admixture, and it provides an avenue to understand TB susceptibility. So to do this, we have this Northern Cape TB project ecosystem, and it's this huge multi-institutional collaboration of different scientists applying different modes of understanding what shapes active TB progression, from the epidemiology to single-cell biology to genomic studies. Today, I'm only going to show a sliver of this work. First, we're going to talk about socio-demographic risk factors. So how does access to education or lifetime migration between rural areas and towns affect progression to active tuberculosis? Next, we're going to zoom into the immune response of tuberculosis. So when we look at the immune cells, how are they responding between latent and active TB infection? And then we're going to, for future direction, we're going to talk about how we can integrate genetic variants into this, understand how genetics affects the immune response. So the roadmap for this talk is broken into two parts. First, who is at risk at TB, and then what cellular immune processes differ in disease state. And then we're going to zoom out of TB and South Africa and think more broadly about African genetic diversity and how we can represent the full spectrum of people descended from Africa. Okay, that brings me to my first water break. All right. So I'm going to first start with data collection. So here the phenotype is lifetime active TB progression. So in your lifetime, is there any microbiological evidence of converting to active TB? So from PCR tests to history of antibiotic use, we have access to their medical records here. So we're collecting saliva for genotyping. We collected saliva for genotyping, biometric data, a bunch of health records as well. And as Brenna stated, this started before I joined the program in 2017. This project is still ongoing up to now. I think we have close to 5,000 people now. By the time we had collected a little over 1,000 people from 12 community health clinics in South Africa. So how we're defining cases and controls here, all our cases are HIV negative individuals because we don't want HIV to kind of confound why the progression to TB with any microbiological evidence of MTB infection, and they're all adults as well. And our controls are participants with no medical history of active TB. So not only do we take self-report, we have to like scan through all their medical records as well to ensure. And so who is more likely to progress in this community? So we are implementing a few models to do this. One, the common risk factor model. So there are a bunch of variables known from the literature like education, gender, smoking, alcohol. And next, we're going to do a migration model. So how does lifetime migration between a town and a rural area affect progression? And this is really just an extension of the first model, but we're adding an interaction effect between residence and birthplace. So were you born in a rural area or town, and did you migrate to a rural area or a town, or did you just stay in the same area? Finally, we're going to run a socioeconomic model, and this is also an extension of the common risk factor model, but now we're adding an interaction term between age and education. So some of our main findings, overwhelmingly, males have 3 times the odds of active TB compared to females. And this result has been shown in multiple South African, African, and global populations as well. And to some extent, it's a mix of both gender and sex. You know, sometimes males may have different like healthcare seeking behavior, but this could also be some sort of like hormonal or genetic difference between males and females. And it's not until we zoom into the genomics, you can understand what else is causing this. Next, as expected, we see individuals that reside in towns, so higher population size, have 2.5 times the odds of active TB in this population. And this was published a few years ago in PLOS Global Public Health. So continuing that, these are the results of the socioeconomic model. Here, our x-axis is measuring years of education. And at the time, years of education was our best proxy for socioeconomic status. So the more education you have, we think that's capturing like access to healthcare and less overcrowding and behavioral practices that are protected. And it's color-coded by different age bands. So at lower ages, younger age bands, the more years of education you have, the lower the odds of active TB. But in older ages, this pattern inverses where the more education you have, the higher the odds of active TB. So what we think we're capturing here could be a few things. Apartheid was abolished in 1994, and access to education and healthcare. People that finished education during apartheid and before and after had very different outcomes. So the kinds of jobs that were available may have been jobs that are involve more community exposure. But another thing that's also discussed in the manuscript is that this could be a signal of survivor bias. So we are only able to sample old people that survived tuberculosis, and the people with a lower socioeconomic status that died, we are unable to capture them. Next, this is the main result from the lifetime migration model. So the x-axis here is measuring where our participants born in a rural area or town. The y-axis is the odds of active TB. And then it's color-coded if they currently reside in a rural area or a town. So for this blue box over here, if you're currently living in a town, regardless of where you were born, your risk of TB is elevated. Next, individuals born in a rural area and currently living in a rural area have an attenuated risk of TB. And most interestingly, this was not what we expected at first. Individuals that were born in a town, so higher community exposure, and then moved to a rural area, have the lowest odds of active TB. So one thing we think could be happening here is that people that move from, they're born in towns and then move to rural areas, we have accrued wealth or have better access to education and healthcare, and then they take those protective effects to a rural area. If you want to learn more about the study, other results and limitations and discussions, please check out my paper from 3 years ago. So epidemiology gives us the risk landscape, but not the mechanism. So we can see that active TB risk is patterned by gender, residence, and historically embedded socioeconomic context. But these variables are telling us who is at risk, but not what immune programs differ in disease states. So next, I'm going to talk about a technique called single-cell RNA sequencing that lets us zoom inside the immune cells and see what's happening in tuberculosis, which brings me on to the second section. So data collection was a bit similar to the first part, where we're collecting saliva, biometric data, and health records. And here, our phenotype are recent TB progressors, and I'll talk more about that in the next slide. We're also collecting blood to capture the immune cells this time. And data collection started in 2023. We collected 187 people from 3 community health clinics. And for this part, I flew to South Africa for most of my third year to start data collection. So here, our phenotype is a bit sharper than last time. Instead of a lifetime evidence of microbiological infection, we want people with a positive TB PCR test within 7 days of getting positive, and they have to have been on antibiotics for a very short time. And we're doing this because we want to capture like the truest state of an infection rather than letting the signal get attenuated by antibiotics. And our controls, after filtering down, we had 73 people. They're all HIV negative with a negative PCR test as well. And then the nurse we worked with also checked them for flu and COVID symptoms at the study. And we asked them if they had had the flu or cold within the last 2 weeks. And we're doing this as well because respiratory illnesses may share similar biological pathways. So we want to get the cleanest response as possible. All right, so what immune programs are different between those people? This is how a sliver of the data we collected looks like. It's similar variables to the previous study, roughly equal numbers of baseline controls. There's still a significant gender difference as well. So we did recruitment, and from the field collections and the sequence data, my computer was a really, really long, arduous journey. And anyone that saw me at Sophia's knows that I was always talking about like this project. So I really want to thank here my collaborators, the Muller Lab in South Africa, who sent me in Cape Town, the Yi Lab at UCSF, and Solomon Lab, who's visiting from UCSF as well, that helped me with a lot of the wet lab work. So why single-cell RNA sequencing? I'm going to start by explaining the more common technique called bulk RNA sequencing. So if we imagine a tissue like blood, like a smoothie, it's like a mix of all of these different cell types. You want, and you do a bulk RNA sequencing, it gives you like average gene expression. So if there's any sort of cellular heterogeneity, which there usually is, it's going to be masked. But with single cell analysis, you're able to tag and barcode each individual cell, and so you can get distinct expression profiles, and it reveals heterogeneity and subpopulation structure. So that's what we're going to be using for this talk. And we're using an extension of this called CiteSeq. It's a really long name, but what you need to remember here is that we're able to tag the cell surface proteins as well. So we have a panel of the 131 antibodies that we're using to confirm the cell types. All right, so I'm going to introduce you to the cells I've been working with. These are called peripheral blood mononuclear cells. I like to call them liquid gold as well. So this is how they should look like after they've been spun very carefully at the right speed. this is what they should not look like, which is how, so this is red blood cell contamination, and we don't want red blood cells, we only want the immune cells. And this was my first time running a centrifuge in like 10 years, and it didn't work. But one of the reasons for that was it's just hard in rural areas to get all the technology you need. And the centrifuge we had been working with was really old. We couldn't get an engineer to fix it, so I tried to fix it over WhatsApp video. And I was doing that. I made it even worse. Yeah, we're able to, it took about 6 weeks of trial and error, and we finally got a good running centrifuge. So for the rest of the talk, I'm just going to call this like blood. And we're going to break down the blood into two major components. One, the innate immune system. So we can think of the innate immune system as like fast responders, very inflammatory, and those will be composed of monocytes, natural killer cells, and dendritic cells. Next, the adaptive immune system. So they're more like target specific, and they have a strong memory response. And we break them down into T cells and B cells. And so these are like major lineages, but all of these cells have minor subtypes that we'll go into only a little bit. And there are other, there's another group that are like quasi-innate, and they're a mix of both, but we're going to ignore that for this talk. So now we have, we've done all the field work, the wet lab work, we have the sequence data, and we can imagine this as this like high dimensional matrix. So you have thousands of genes, thousands of cells, and then we want to reduce that to a 2D space to visualize this. So I'm using the Surat package for single cell genomics, and I'm going to show you what we call a uniform manifold projection of the UMAP. So if you're familiar with the PCA, it's kind of the same. We actually use PCs to make the UMAP and a bunch of other clustering algorithms. But basically, it helps preserve both like local and global structure. So the cells that are clustering together are similar transcriptional profiles. So it's going to look something like this. So when we apply that to our data, we have 500,000 cells after processing. This is a really huge number for anyone who does single cells. So we have this huge data set, and this is what our data looks like. It's broken down into the innate and adaptive immune system. And we can zoom in and see those 5 major lineages we spoke about, and the other cell type we ignore for now. And then we can zoom further in, and then we have over 15 different cell types from the major lineages in this UMAP. And for this, I really have to thank the Solomon Lab once again. I've been going to the lab meeting over Zoom for about 2 years, and like for the first year, we're just like going through every single cell type, checking what antibodies are being expressed, and it was very like arduous work, but we got there, and we leveraged the cell surface proteins that we talked about earlier. So now we feel confident about our cell types. Let's start asking some interesting questions. So first, I'm interested in immune cell abundance. What immune cell populations are expanding or contracting upon infection? Differential gene expression. So within each cell type, which genes are changing? So upregulated or downregulating in disease states. And then making sense of these differentially expressed genes, what biological pathways are being activated. And for future directions, a statistical model called EQTL mapping, thinking about how genotype acts differently by context. So what we know about TB right now is that the immune landscape is very heterogeneous and inflammatory. So the plot over here on the right, the x-axis is capturing different stages of TB from uninfected all the way till they finish their antibiotic regimen. The y-axis is measuring abundance, and it's color-coded by those 5 major lineages. And we see that some innate, so those first responder fast cells may expand or become more inflammatory. And the long-term adaptive or killer-like cell types may become depleted, activated, or show exhaustion-like signatures. And so for the first question, thinking about cell composition, if you imagine this like orchestra of different cell types with different functions working at a baseline level, upon infection, how do they expand or contract? So these are our results for this. It's color-coded by cases and controls. and I've binned them into the major age groups. You don't need to know all the cell types. The first panel is showing major cell types, and the second panel is showing the more rare cell types. So on average, those like adaptive immune cell subsets are getting depleted, and the innate immune cells are getting enriched apart from the dendritic cells. So we've done cell abundance analysis. Now I want to see, like, within each cell type, do cells occupy a different transcriptional states between latent and active TB? So when I run this across all the cells, I'm just showing a subset of the cell types. We see that for some cell types, there are hundreds of genes being upregulated and downregulated. For some cell types, there's not a lot going on. So we're going to zoom in on one of the cell types monocytes and look at the volcano plot. So here, the x-axis is showing the log2 fold change, and it's each circle is a gene, and it's color-coded by significance level. So any red dots we see on the right are genes that are upregulated with people with an active TB infection. The circles on the left are the ones that are upregulated in healthy individuals. And we can go through, if we might go through a few of them, we can see from previous papers, all of these genes have been found as immune response markers for tuberculosis. So if we want to try and create a working model of what is the relationship between abundance shift and differentially expressed genes, we can bin them into different groups. So here the x-axis is going to capture the abundance shift. So the first analysis I did for this section. So are the cells decreasing or increasing the TB? And then the middle part is if there's no significant shift. And the y-axis is going to show the number of differentially expressed genes. So the size of the circles you're going to see correspond to the mean proportion of the blood, and it's color-coded by the major lineages. So we have these minimal responders. They are a mix of the innate and adaptive immune system. So they are not expanding or contracting, and there are a lot of differentially expressed genes between them. So not a lot going on. We have these abundant responders. So these guys, they are getting depleted upon infection, but they don't have a lot of differentially expressed genes. So the transcription landscape isn't changing in a dramatic way. Next, also all these names, I'm creating them myself. We have these like state shift responders who they're not expanding or contracting, but they're having huge transcriptional changes. So they're having hundreds and hundreds of differentially expressed genes. And last, we have these major responders. So the state shift responders are all adaptive T cells, and then the major responders are innate T cells. And this makes sense that the innate T cells act very fast and very quickly. So they're the ones that are expanding and having lots of genes being upregulated. So next, we still have hundreds and hundreds of genes. So we could either like go through each gene and Google each one, which will add an extra 2 years to my PhD. Or we can think of a way to systematically analyze like a ranked list of genes. So I'm going to do an analysis called gene set enrichment analysis, where you basically feed in a ranked list of differentially expressed genes for each cell type, and it basically provides an enrichment score for what biological pathway is significant. There are going to be a lot of rows in the next slide. So here are results for controls. Here, each row is a biological pathway that very smart molecular biologists have, assigned and created. And each column is a cell type, and the top boxes break them into those like major immune clusters. And on average, these processes that are enriched are protective and baseline regulatory processes. So I can highlight one, for example. This is called TNFA signaling via NF kappa B. And basically, it's basically saying that people that have the latent, so healthy individuals that are enriched for this biological pathway are not progressing to active TB. An interesting thing about this pathway is that drugs like Humira or similar ones are TNF inhibitors. So if you are on medication or have family medication for things like rheumatoid arthritis, a side effect of this drug is tuberculosis, because this pathway is actively being blocked. So next, we're going to show our cases. I know there are a lot of pathways here as well. Once again, each column are those unique cell types. Each row is a biological pathway. And I've already said this earlier, the intensity of the blue or the color is how enriched that pathway is. So we're not going to go through like every one by one, but we did do that with our collaborators. But what we're seeing here is that in active TB, inflammation takes over. So all the pathways that are being enriched are things like interferon, gamma, and alpha response, and also things like oxidative phosphorylation. So the mitochondria energy metabolism is also changing in a dramatic way. And this is happening across multiple cell types. All right. So those were 3 questions I answered. I'm going to walk you through the next section of things I'm going to do this summer, thinking about the how to combine genotype and gene expression in the same model. So studying the genetics of TB is important because multiple studies from twin studies, innate inborn errors of immunity, and genome-wide association studies have estimated heritability of TB phenotypes to be anything from 15 to 80%. So there is some heritable component of TB phenotypes. And here we're going to combine single cell expression data, which I've walked everyone through, with whole genome sequencing. So we recently finished the variant calling for our whole genomes a month or 2 ago. We have millions of variants. We're very excited about that. And we're basically going to combine them together. So in this schematic, the x-axis is measuring gene expression, which is what we were looking at earlier in this talk. And we can imagine for every genetic variant, there's like within this room, people are going to be bid into like 3 different genotype classes. So here I'm interested to see upon response to infection. So the black ovals are representing tuberculosis. How does gene expression change in respect to the genotype? And these loci were interested in our term EQTL. So that's what we're doing EQTL modeling. So if you're familiar with GWAS, it's like a very similar model to GWAS, but here your phenotype is gene expression. And so we're also going to leverage admixture for these genotype-phenotype associations. Here is another admixture plot. This is for our TB participants from the transcriptomic study. We see once again they're a mix of multiple ancestral groups. So we have the Komani San, so those indigenous hunter-gatherers in blue. We have West African, Indonesian, and European individuals as the source populations. And we can see the Northern Cape people are a mix of all of these populations as well. And so admixture can be a double-edged sword. If you don't control for it, you can get false positives. But then we also want to see if we can interpret whether signals reflect any sort of systematic association with genetic ancestry. So we're really excited to see where this work is going to go next. So the take-homes for the TB section, TB progression is patterned by both social and epidemiological contexts. Active TB is associated with cell type composition changes and immune gene programs. And together, these layers are motivating genetic analysis of the immune response. So now we're going to zoom out of tuberculosis and South Africa and think more broadly about interpreting genetic variation. And for this, I've had the pleasure to work with 2 really amazing undergrads who are both graduating. Majestic is a human biology major. Trying not to get emotional. Wait till you see the acknowledgements. Majestic is a human biology major with a minor in African American studies. And Mustafa is a double major in anthropology and data science. So to start this, we're going to travel back to Nigeria in a different context. 3 years ago, I stumbled upon this paper. So many Nigerians. Why is Nigeria overrepresented as ancestral genetic homeland of legacy African North Americans? So I did think this was slightly inflammatory for a title, but the author of this paper actually gave the STAR lectureship about 2 to 3 years ago, Fatima Jackson. But to translate this in layman's terms, she's really pointing out this convenient shortcut in population genetics. Many studies use the Yoruba people from Nigeria as a stand-in for African ancestry for people out of Africa. But we know that African and Afro-descendant people reflect multiple source regions, population histories, and ecological backgrounds. So, for example, here is a schematic of all the different areas of the slave trade, and it really ravaged not only the Yoruba people of Nigeria, all over West Africa, parts of Eastern and South Africa as well. So when you think about the descendants of the slave trade living in the Americas, they're really a mix of all of these populations shown on the map. So I'm going to walk you through a schematic of how doing this in the wrong way can be problematic and how we interpret signals of selection. So over here, we're showing a Manhattan plot. So this is not a GWAS, I promise. This is a selection scan. The x-axis is capturing genomic positions, so SNPs. The y-axis is significant p-value, and any circles above the horizontal line are SNPs that are significant. So what a selection scan is doing in layman's terms, it's looking at significant allele frequency differences between two populations. So a source population on the right, so here we're assuming it's just the Yoruba people from Nigeria, and then descendants being African Americans. And so when this was done, we see that there's this like SNP in CD36. CD36 is a fatty acid metabolism gene. So we think that this SNP is being selected for. And so there's a significant allele frequency difference. But when the authors redid this analysis using a broader set of West African groups, the signal disappears completely. So when I read this paper, this was a huge motivating factor to create this chapter. And so when we don't have the right set of reference population, it's very easy to do data storytelling. So this paper came out last year. It found that actually a position in this SNP is implicated with dilated cardiomyopathy in African Americans. And we also know this SNP, this gene is involved in malaria resistance in Africa. So we could think maybe there's some sort of antagonistic pleiotropy. This gene involved in malaria resistance is now affecting cardiomyopathy in African Americans. But really, sometimes selection signals are just reflecting methodological biases. So when we're comparing an admixed population to like the wrong reference population, what we think is a dramatic frequency difference may just represents population structure within the source populations. And so this is the motivating story for Kappa Web, how better population representation leads to better biological interpretation. So Kappa, this is a really long name. I'm not going to read it out, but it's basically a huge consortium made of populations from all over the globe. It's high coverage whole genome sequencing data. We've also processed 39 million genetic variants from close to 4,000 individuals from 62 global populations and 12 broad ancestry groups. So the map here is showing a sliver of the populations we work with. We also have populations from Europe and Asia and indigenous Americans as well. So, we've talked a big talk. We have this huge whole genome sequencing data from all over the world. Now I want to see, do these populations in Kappa sufficiently capture the common genetic variation in African descent populations? So for doing this, I'm turning to my colleague James and Graham Coop from CPB, who came up with this amazing blog post and visualization tool called Euler diagrams. And so we can imagine, so I actually lured James to the Genome Center, I think last year, spring break. I didn't let him leave till he taught me how to do this. So if we have like a genomic sequencing data set, we can imagine an area of the ellipse to represent number of common genetic variants. And so this is representing all the common SNPs in an admixed population like African Americans. And so what we can do is we can overlap the different putative, what we think are the putative source populations. So the overlap between the ellipses represent the shared genetic variation. And the ones that don't overlap are the private in quotes, because no SNP is truly private, are the ones that are private to the populations of interest. And so when I applied this to the admixed populations, we see a few things. So this is applied to real data from the Kappa data sets with our admixed population being African Americans, and all the other populations are what we think are the putative source populations of African Americans. So each broad ancestry group is composed of multiple subpopulations within them. So we see that majority of common variation is shared across all populations, which is expected. But if we zoom into that blue box there, that light green color are the amount of SNPs that are private to African Americans. And because this is such a small proportion, we can see that the source populations we want to use to model admixture are a pretty good data set. So this is great. I said that already. And so now I'm going to walk you through what this web tool looks like. This is the landing page. So if you're reading a paper and you see a SNP like CD36, you would like pick your genome assembly, pick your chromosome and position, you search it, and automatically it gives you a list of information. So it tells you what gene it's found, and it also shows you different like metrics. So like the GURP score is measuring evolutionary conservation. So it's natural selection preventing mutations in that region. If it's a protein coding SNP, it also has biomedical and molecular like pathogenicity scores. So is a SNP damaging or tolerable? Then it computes allele frequencies across all 62 populations. So if we stick to that CD36 SNP, we can see that across, so the green is West Central Africans. Across West Africans, there's so much heterogeneity in the allele frequency. So whatever it's going to be, whatever, if you're doing a selection scan, whatever source population you choose is going to change the results. So you really want to use a weighted combination of what we think are the true source populations. And I also want to emphasize, this is also an interactive tool. Because there are so many populations, you can just click and choose what populations you want to see, and it automatically visualizes that. And thank you, Mustafa, for like all your hard work. It also shows population structure. So by this, we're measuring FST, so capturing differences in heterozygosity. It's also interactive. So whatever population you're interested in, you just click and it automatically gives you a heat map measuring FST. And lastly, it's going to show shared variation as well, which are the other plots we talked about. So this is what I think Mustafa calls this an entropy diagram. So this shows what our whole pipeline looks like. We coded this in a bunch of different languages, and we carried out the computation on the whole genome. But unfortunately, our full data set is too big for Google Cloud Run. So what I did was to subsample it to only protein coding SNPs. So that's 1% of the genome. That was still too big to deploy Google Cloud Run. It worked, but it was really buggy. So we kind of revamped this to Kappa Web version 1.0, where we are focusing only on evolutionary and medical relevant traits among African ancestry individuals. So this website is now live, and so that's the barcode for the website and the barcode for the SNPs of interest. And this works on Google Cloud. What a great call. So future directions, we're working with collaborators in Quebec at Cartagena to migrate our whole web infrastructure to Cartagena so it can host the computation of all 39 million variants. And I really want to emphasize, as sequencing is getting cheaper, the real issue here is storage. And me and Brenda can talk to you about hours of like how much has been spent talking to IT about getting where to store our data. So conclusions from this section: African ancestry is not a single genetic reference. Our reference panel charts can shape how we interpret apparent signals of selection, and Kappa Web can be leveraged both as an educational and research tool for evolutionary and population genetics. Right. So we're going to do questions first, and then we'll do acknowledgements. I'm going to try to not cry.
Lovely talk, great work. So tuberculosis, we know it's a lung disease. How much can we verify study?
Great question. I have two answers for that. So the blood really is a mirror of what happens in other organs. That's why when you go to the clinic for like vitamin levels or liver enzymes, the blood is used. So it really mirrors studies on like non-human primates and rats have shown the cell signatures and abundance changes that happen in the blood are somewhat similar to what happens in the lungs. And the other practical reason is that it's hard to get lung tissue samples from people that are alive. So, great question.
Yes. Yeah, it was really cool. I appreciated all of the different angles you took answering these questions. Something I was interested in hearing you, I guess, speculate about a bit is, or to sort of say why you chose to do this, your decisions to only focus on individuals that were HIV negative. I'm wondering to what extent you think that there might be interactions with how, what your results would show if you were instead looking at HIV positive individuals, or why you did that subsetting.
Yeah, that's another great question. So HIV has such a strong inflammatory response that for those pathways that are shared, it would be hard to know if the inflammation response, like from the gene set enrichment analysis, is HIV or tuberculosis. So people with HIV have like 20 times the odds of active TB. So we want to kind of like clean out the noise from that. But there is an interesting phenotype called like resistors, where there are people that are have HIV and then they do not convert to active TB. So that's a really interesting phenotype to kind of understand what makes people like not get sick. Just finding those individuals are like really hard.
I have a basic single cell RNA question.
Yeah.
Also, really impressive to lay out the foundation for all of these spin-off studies. Like, this is great. When you do the single cell RNA, are you also capturing activity of TB? If someone like, is that like bioinformatic trash or whatever? Or do you just save that, or can you look at that, or how does that work? Or is it just human DNA do you get from single cell?
That's a great question. I'm gonna give you like a side answer to that, because Brennan had this interesting idea of seeing if we could capture, almost do like a pseudo HIV test from this, where we're like, if we could align our transcript reads like the HIV genome, if there are alignments, we could assume that HIV was captured. So we could do the same thing. But in terms of like seeing like the actual like tuberculosis pathogen, I am not able to do that. But like, because we are seeing inflammatory responses that match the literature, we assume like if like interferon alpha is being enriched, then MTB is there, but I'm not like physically seeing the pathogen.
That makes sense. Thank you.
Grace, yes.
So with an active TB infection or any active infection, as you said, you'll see a really elevated inflammatory response. With the non-active TB, or let me backtrack, with exposure to any infection, that will also kind of elevate different immune cell lineages.
Yes.
One, was your PCR test for the TB, like active TB infections, does that also include like exposure, or is it just a current active infection? And two, how, if you did have exposure in like your control group, how do you think that would actually manifest in like the different tests that you did?
Yes, great question. I also want to mention Grace is in the vet school, and I used a bunch of her immunology notes to study when I was like trying to delve into immunology. So everyone that's latently exposed, that's latently infected is exposed. So by the age of 35, 85% of the population is exposed. And so we do see inflammatory response in the latent people as well, but the cell types are different. So if you have latent TB, so these cell types called monocytes get activated into a macrophage, and they form this cluster called the granuloma, where they kind of surround the MTB pathogen. So they're keeping it at bay. And so I believe that inflammatory response are those like cells holding in the granuloma. But the inflammation response we see in cases are the interferon gamma responses. So there's inflammation in both, but the intensity and at what cell type is different. Oh yes, yeah. So there are two tests for TB. There's the Gene Expert, which is like the PCR test that's showing if you have that pathogen currently residing. And then there's the IGRA test, that's an interferon gamma response assay. So it's a blood test, but it can't really differentiate. It shows you like, it reveals that you've been exposed to TB, but it can't differentiate if it's like active TB or latent TB.
First of all, great talk. It really amazed the breadth of your research. So my question really deals with like the last part, this new like really innovative tool you guys kind of created, Kappa Web. And because I guess my question is how translatable you can use it as a tool to study non-pulmonary conditions. And because you're using a kind of like a reference data set that's geared towards studying asthma in African populations, but can you apply it to like maybe looking at neurological disorders or things like that?
Oh, great question. And I wasn't clear on that. So like Kappa started out as like an asthma project, but after the first few years, it just expanded to just like African genetic diversity. So it's just like sampling people from Africa, and it's phenotype agnostic, but like the name just kind of stuck over the years. So right now we have malaria, lactose persistence, blood and cardiac disorders, height, and there's one more I forget, but we have about 8 phenotypes that are represented right now. But really I have the whole genome computed. I just need like data store that can let me like deploy everything at once. Yeah.
First of all, this is incredible, Oshiomah. Me and Oshiomah know back 20 years ago, we were both in the same high school. And so this is incredible, bro. To just see you speak like this, I'm just like, what the heck? So real. So I'm more of like an engineering major. So most of these things are like, this is crazy. This is good stuff. But I think my question is like, what does this mean for people who are in a high risk community? What does this research mean to them? In a sense.
That's a great, it means a bunch of things. So one thing that's really important to our lab is community feedback. So when my first study was published, our lab manager, all our students, we created different like health pamphlets translated to like Afrikaans to kind of show like the results of the study. So like we always package the results in the local language dispense. And the second thing is, so when I was in South Africa, my paper had already been published like a year or two before, the Brenna had already like dispensed the results, but there was a different study that was looking more about genetic ancestry and like the source publisher of the slave trade. And if you remember, my very first amateur slide, we used both East Asian and South Asian populations to represent like the source populations of like people in South Africa. But my lab mate Dana found that it was really people from like Indonesia that were like the source populations. And so me, Dana, and another collaborator in South Africa, we went to like local high schools. We had like posters in like community centers and like libraries and museums, like explaining like the results of our studies.
That's awesome, man. It's really good.
Yeah.
I have a question about, so you mentioned that what you're really limited on for the Kappa Web is storage.
Yes.
Is that a feasible amount of storage that you can obtain, or is that like still going to be a limiting factor, you think, for like the long term of this project?
I think we can obtain it. So like this, like these Cartagena people in Quebec, they have like a huge web infrastructure and really using Brenna's connections to do that. But yeah, it's only like 30 gigabytes. But like doing some of those computations on the fly is just like really memory intensive. Yeah, but it is doable, and hopefully in the next few months we can get it deployed. Right, there are no questions left. Acknowledgements. So first, I want to thank all the participants that have donated their time and energy, data to the study. We really could not do this without all of them. I want to thank all our nurses and field research assistants, especially Thelma, who I worked with 2 years ago and has now passed away. And I always promised I was going to
Shuma,
visit her
take it
when I
easy.
went back to South Africa, but she's no longer with us. I want to thank the Hen Lab. Greta, you've been an amazing mentor over the last few years, and I really came in with no bioinformatics, barely any coding experience. You gave me a chance, and I think we came up with a really good body of work. Every single person here, past, future, and present. I want to thank the Suleiman Lab. I have really been just like this huge screen on their Zoom monitor for like 2 years for like letting me ask you all the really dumb immunology questions and helping me with the wet lab work and helping me like think more like an immunology. So thank you so much. I want to thank the Moore Lab in Stellenbosch for hosting me for 6 months and all the friends there I made. I want to thank my cohort. We started in the pandemic and with forest fires, and we survived. We were wearing masks for like 2 reasons. We didn't want to get sick, but like just seeing how we stuck it out over the years, and we're all graduating this year. I'm just really grateful to have spent the last 6 years with you guys. More broadly, all my friends from Pop Bio, I love you all. You guys are amazing. And then all the other friends I made all along the way. Including my best friend Bahati, who came here all the way from Providence. Thank you. I want to thank my family. My parents are the most hardworking people I've ever met. My parents grew up selling food on the street. So this is just such a big deal to them. So I'm really grateful. My siblings are all super smart. They're all younger than me, but they've been working their whole life, sort of since undergrad. They've also helped me like financially as well. So thank you all. And everyone else I couldn't fit on the slide, my decision committee, Brenna, Graham, Joel, and Sarah, you guys are all amazing. All my funding sources, of course. Margaret, the smartest statistician I have ever met. My decision committee, the E Lab, Austin Reynolds, and the Kappa Consortium. So thank you all.